Ladies and gentlemen, esteemed colleagues and medical students, today we delve into the fascinating topic of gestational diabetes mellitus, GDM. GDM is a complex condition that affects pregnant women, characterized by high blood sugar levels. It poses a significant risk to both the mother and the developing fetus, with potential long-term implications on their metabolic health. Now, let us embark on our journey of understanding GDM in depth, exploring its various aspects, from hormonal changes to diagnostic criteria, complications, genetic factors, beta cell function, insulin sensitivity, glucose transport mechanisms, association with obesity, impact on offspring's health, epigenetic modifications, future cardiovascular risks, and finally, novel treatment options and preventive strategies. GDM is primarily attributed to the action of placental hormones, which lead to an increase in insulin resistance. These hormones, including human placental lactogen, progesterone, and cortisol, interfere with the normal action of insulin and contribute to the elevation of blood glucose levels in pregnant women. Accurate diagnosis of GDM is crucial to ensure timely intervention and appropriate management. We will discuss the various diagnostic criteria, such as the glucose challenge test and oral glucose tolerance test, and understand screening methods used in clinical practice to identify women at risk for developing GDM. GDM can lead to a multitude of complications for both the expectant mother and the developing fetus. We will explore the increased risk of preeclampsia, gestational hypertension, preterm delivery, macrosomia, excessive fetal growth, birth injuries, and the impact on future health outcomes for both mother and child. Research has shed light on the genetic factors involved in the development of GDM. We will delve into the specific genes and genetic variants associated with GDM susceptibility and explore the interplay between genetics and environmental factors in the onset of this condition. During pregnancy, the beta cells of the pancreas undergo significant changes in their function. These changes are essential for proper insulin secretion, a hormone that helps regulate blood sugar levels. The physiological adaptations that occur during pregnancy can sometimes lead to a condition called gestational diabetes mellitus, GDM. GDM is characterized by elevated blood sugar levels during pregnancy. Understanding the alterations in beta cell function can provide valuable insights into the development and management of GDM. Let's explore these changes in more detail. During the first trimester, insulin sensitivity generally remains relatively stable. However, as pregnancy progresses, insulin resistance gradually increases, particularly in the second and third trimesters. This physiological phenomenon is thought to be influenced by hormonal changes, including the secretion of placental hormones such as human placental lactogen and progesterone. The decrease in insulin sensitivity during pregnancy is necessary to provide adequate glucose supply to the growing fetus. However, in some women, this insulin resistance becomes excessive, leading to gestational diabetes mellitus, GDM. Understanding the dynamic changes in insulin sensitivity throughout pregnancy is therefore crucial in diagnosing and managing GDM effectively. Understanding how glucose is transported from the mother to the fetus via the placenta is vital in comprehending the pathophysiology of GDM. We will explore the intricate mechanisms involved in glucose transport and their potential dysregulation in the context of GDM. The prevalence of obesity in pregnant women has been steadily rising, and it is closely associated with an increased risk of GDM. We will investigate the complex relationship between GDM and obesity, exploring the underlying mechanisms and potential strategies for prevention and management. Gestational diabetes mellitus, GDM, a condition where high blood sugar levels develop during pregnancy, can have significant impacts on the metabolic health of the offspring. Research has shown that children born to mothers with GDM are at an increased risk of developing obesity, insulin resistance, and type 2 diabetes as they grow older. These long-lasting effects on metabolic health can be attributed to various factors, including genetic predisposition and exposure to high blood sugar levels during prenatal development. Understanding these risks is crucial in managing the health of both mothers and their children. Gestational diabetes mellitus, GDM, is a condition characterized by high blood sugar levels during pregnancy. Recent studies have suggested that certain modifications to the DNA and proteins in the body, known as epigenetic modifications, may play a role in the development and progression of GDM. These modifications include changes in a process called DNA methylation, as well as alterations to proteins called histones. In this section, we will explore the latest research investigating these epigenetic changes and their potential implications for GDM. Research has indicated a growing body of evidence that gestational diabetes mellitus, GDM, can elevate the likelihood of developing cardiovascular diseases in both mothers and their offspring. In this section, we will delve into the intricate mechanisms that connect GDM with future cardiovascular risks. Additionally, we will explore various prevention and management strategies that can be employed to mitigate these risks. Understanding these multifaceted connections and implementing effective strategies is crucial for the long-term health of both mothers and their children. These innovative approaches include behavioral modifications, such as implementing a healthy diet and regular physical exercise, which have shown promising results in managing blood sugar levels and improving overall maternal health. Pharmacological options, such as oral medications like metformin or insulin injections, may be considered in cases where lifestyle interventions alone are insufficient. Additionally, researchers are investigating potential targets for interventions, such as targeting inflammation or oxidative stress pathways, to prevent the onset or progression of GDM. These emerging therapeutic strategies aim to not only treat GDM but also reduce the risk of long-term complications for both mother and child. While these novel treatment options and preventive strategies hold significant promise, further research is needed to fully understand their efficacy and safety profiles. It is important for healthcare providers to stay updated with the latest evidence-based practices to provide optimal care for women with GDM. In conclusion, gestational diabetes mellitus is a multifaceted condition with far-reaching consequences for both the mother and the developing fetus. By delving into its intricacies and understanding the underlying mechanisms, we can pave the way for innovative approaches to prevention, early diagnosis, and effective management of GDM, ensuring the best possible outcomes for mothers and their children. Thank you for joining me on this enlightening journey through the vast domain of GDM. I hope you feel equipped with the knowledge and enthusiasm to further explore and contribute to this evolving field.